A very good morning, students. So far we were studying about the basis of classifications, and we successfully completed all the basis of classifications regarding the kingdom called Animalia. And today we are going to discuss on an ultra important topics, that is the phylums. Now, when we talk about this phylum, we are going to discuss about each and every phylum in depth. We know the kingdom called Animalia is divided into two kingdoms, two subkingdoms. One is called the non-chordates and chordates. So this after the kingdom. So we had the first one called the kingdom called Animalia, and this kingdom is divided into two subkingdoms. One is called the non-chordata, and the second one is called the chordatas. So these are the two things that we have discussed so far. Now this non-chordates and chordatas are further classified into phylums. So now among this phylum we have 35 phylums are there, and among this 35, 11 phylums are the major phylums that we know. Now among this 11, 10 phylum belong to this non-chordates and 1 belongs to this vertebrata, that means chordates. Clear? So in this class what we are going to study is, first we are going to study all these 10 phylums in detail. So what are all these 10 phylums, starting from Porifera and ends up to Hemichordata. You can recall all these phylums as well as the examples in between. So we go with Porifera, then Cnidaria, then Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, then Echinodermata and Hemichordatas. So all these 10 phylums along with the examples we have already discussed. Okay. So now in these three classes in this week, we are going to concentrate on each and every phylum. So in one class we will be discussing about 2 to 3 phylums in detail, so that you will complete entire phylums within 3 classes. Okay, and then we will concentrate on this chordatas. Okay, right. So now we will go for this phylum in depth, all these non-chordates. So one by one now we will discuss. Now whenever we understand, whenever we study about these phylums, we are going to study 11 phylums. In depth means all the particular characteristics, general characteristic, everything we are going to discuss on these phylums. So it will be hard for us to put it in mind, and even it will be hard for us to recall it. So I will give you a proper way for you to understand. So please concentrate on each and everything. I will help you how to concentrate on, or how to study and how to recall these phylums in detail. Okay, so exam point of view we will study. At the same time, even if you want to recall any phylums immediately, I will show you, I will give you a simple method of remembering those phylums, so that it will help you to recall whenever you require. Okay, right. So our today's topic is, we are going to concentrate on these phylums of this non-chordata. Clear? And now we will discuss about the phylum called Porifera. So this is the first phylum that we are going to study under the non-chordates, that is called the Poriferas. Okay. So now whenever we study about the particular phylum, 3 fundamental things that you have to put it in your mind. Number one is, we have to study the distinct features of this particular animal or particular phylum. So first one is the distinct features. The distinctive features. So what is this distinctive features? That is, a particular character is belong to only this phylum. So that is the specific characteristic of that particular phylum. So distinctive features means the particular or a special character of that particular phylum. So that is what the distinctive features are. And second one that we will discuss here is the specific characters of this phylum. That is the specific, or otherwise called, we will not talk it as specific because distinctive, even we can call it as specific. So here we will have the salient features or the general characteristics. So salient features. Now we have distinct features. First of all, whenever we discuss about this phylum Porifera, you should discuss about this distinctive features. That is the specific characteristic. Second one, you should discuss about all the salient features of that particular species or particular phylum. So what are all the salient features? We have so many salient features that you have to remember for all the phylums. So I'll put it together so that it will help you to study for all the phylums. So the first one is the habitat. So habitat is nothing but the place where the animal live. So that is what the habitats are. And the second one is the level of organizations. Level of organization. Now the level of organization here, we are talking about whether this phylum has cellular level, tissue, organ or organ system. And third one is the symmetry. Here the symmetry means, we already know there are two types of symmetry that we have discussed. One is called radial symmetry, another one is called bilateral symmetry. So when you cut the animal, if it gives you two identical offs, it has symmetrical. If it not has any, or if it does not gives any equal off, it is called asymmetrical. So based on the symmetrical, we are going to analyze this organism. Then the fourth one is segmentation. Segmentation, if the organism is having segment, it comes under segmentation. Okay, so that is called segmentation. The fifth one is coelom. Coelom is a body, cavity. All these things we have already discussed in the basis of classifications. Clear? So we have only the subtopics. All these are the salient features of this particular phylum called Porifera. So whenever we talk about the phylum, we should study about the distinct features, salient features like habitat, level of organization, symmetry, segmentation, coelom. Then we will go for the general characteristic, some physical characteristic like digestion. Then you will talk about the circulation. Then we will talk about respiration. Then we will talk about this excretion. Then sex, or we will call it as reproduction. Then we will also discuss about the fertilization and development. Fertilization as well as development. And finally, we will end with examples. So all these things that we are going to study in one salient features. So whenever you study about a particular phylum, you should talk all these points. So if you remember all these points along with the distinct features and example, the entire phylum will get completed. Okay, so these things that you have to keep it in mind. What are all the salient features like habitat, level of organization, symmetry, segmentation, coelom, digestion, circulation, respiration, excretion, reproduction, fertilization and development, and examples. Clear? Right. So now one by one that we will discuss, and then we will complete this phylum called Porifera. Now we will concentrate on this phylum called Porifera in those three aspects, that is the distinctive features, salient features along with examples. Okay. Now when you talk about Porifera, there are two meanings in this word. Porifera means pore bearing animals. So the animal which has pores on its body is called Porifera. Okay, so pore bearing animals. Now the example, the example for the Porifera are called the sponges. So don't forget it. An example for Porifera is the sponges. Now what is the distinct features? Very important. So we will first talk about the distinct features of this particular phylum. So distinctive features. Right. So now when you talk about the distinctive features, and this is the structure of the sponges. Structure of sponges. It has a small pores on the body, that is why it is called pore bearing animals, and these pores are called ostia. Okay, so the pore is called ostia, and it has a big opening here. This is called osculum. Now through this ostia, water will be entering inside. So water enters through this ostia and enters into the sponges, and it comes out through this osculum. So osculum is a big opening. Okay, and now this structure same, I'll just do it for giving certain cells. So now the same structure, I just put it like this. So now this is having inside a center, a cavity like structure here, and this cavity is called spongocele. Okay, so the cavity is called spongocele. Now this spongocele cavity, in this, after water has entered through this ostia, the water will be entering into this spongoceles, and then only it comes out through this osculum. Now this spongocele will have, this sponges, the body wall will have certain flagellated cells. So cell arrangement will be there like this. So these cells have certain flagella like this. So these things are only meant for this phylum called Porifera. That is what it is called, a distinctive features. So here this is called the flagella. The flagella is for the movement of water. The movement of water. So water moves through this flagella into this ostium, and to the it circulates throughout the body cells. Okay, right. And it has the cells. So it has some different types of cells, because the arrangement is cellular arrangement. That is what we studied. So all these cells are independently working. So we talk about these cells. Now there are cells. First cells we will discuss is the conocyte. The conocytes. Now the conocyte will be present. It is also called as the collar cell. It is the collar cell, and this type of collar cells. So conocyte is a collar cell, and this collar cell is present inside. So it is on the inner body wall. Inner body wall. So where can you find this collar cells or conocyte? It is there inside the body wall. And this conocyte will have a kind of, so this is the cell of the conocyte, and this have a flagella. So this flagella is for, I already told you, the movement of the water inside. So inside this, it is arranged in the, like this you will have. So here you will have this flagella. Okay, so this is important characteristic of this, and it also have some amoeboid cells. Amoebocytes. I'll just draw for you to understand this same. So this is the osculum, I told you, and this is the body of the sponges, and this is having the ostia, and this is osculum that you know. Then center it is having the spongocele. Okay, and here you will find some cells inside, and those cells are called amoeboid cells. This is the amoebocyte, and this amoebocyte will form a kind of spine like this. So this amoeboid cell only will form a spine like this. Okay, so this is called the spicules. So spicules are presented here, and these spicules are made up of calcium. This is made up of calcium. Okay, so important thing that you have to understand. So everywhere you will find the spicules inside, outside, everything, and the spicules are made up of these amoebocytes. Okay, so that is what important regarding this. So amoebocyte will secrete a kind of a calcium spicules, spine like structure. Okay, so that is the salient features. Now all this, so everything is involved in the movement of water, like ostia, spongiocele, osculum, as well as the conocyte, everything is involving in it. Now the conocyte will always involve in, it is there inside the inner wall. Outside only we studied the pinococyte cells, already I have discussed in the previous video, you can watch it. Now this collar cell or conocyte will always involve in intercellular digestion, because this sponges do not, or porifera do not have the digestive system. So inside the cell digestion takes place. So it is called intracellular digestion. Okay, so this is very important regarding this salient features. So everything comes under transport of water or transport of water. So this is called the water canal system. So we call it as water canal system. Okay, so in water canal system we got this ostia, then spongiocele, osculum, as well as the conocyte, which is having the flagella. So this is the distinct features of this porifera. Okay, so all this thing that you have to remember, only if you remember this, then we can go for that salient features. Okay, so I hope you understood what is the distinct features. You can find only this distinct features for the sponges and not for any other organisms. Now we will study about the salient features. Now when you study about the salient features, I will tell you a word to remember, so that by seeing that word, those words, easily you can identify or easily you can study all the salient features. So what are those words? That is porifera, porifera match. Okay, so porifera match. If you remember this word, entire salient features you can easily study. All this 11, excluding that example, 10 salient features that we have. So all this 10 you can easily remember. So in this match, this T alone, you just leave it. Okay. So other things will be equally matching all those salient features. Okay. So remember porifera match. Now, so each word that we will circulate and we will study. So what is P stands for? P is for pore-bearing animals. So organism which has pores are called the pore-bearing animals. So P stands for pore-bearing animals. And the second letter is O. So O stands for ostia, osculum. Then you also remember spongiocele. Okay, then everything will be interconnected with a cell called conocytes. Okay, everything is interconnected with conocytes. So what is the role of this ostia? Through ostia, water will be entering into this sponges. Water enters, and this water, when it is in where this ostia is, it is on the lining of the body wall. So if you talk about this sponges, the sponges will have this ostia close to this body wall. Okay, then it will enter into the spongiocele. After water enter inside, it enters the spongiocele. Then water will be circulated through this and it comes out to the osculum. So all these are called water canal system. Very important. Water canal, water will be connected in the form of a canals. Enter inside and comes out. So water canal systems. So through this, what is happening is, through this, food will be gathered. So all this canal systems, gathering of food and circulation will take place, respiration, then we have excretion. So everything is taking place through this water canal system, that is ostia, osculum and spongiosum. So when you remember O, you remember ostia, osculum, spongiosum. Clear? Right. Then the third letter, that is R. So we completed P, we completed O, then we are going for R. So R is for reproduction. You remember reproduction of the sponges. So what kind of reproduction that sponges are having? Actually, sponges are hermaphrodites. In the sense, ovum and sperm are present on the same individual. So they are called hermaphrodites. Clear? So in this, there are two types of reproduction will take place. One is, whenever you talk about sponges, you remember two reproductions. One is called asexual reproduction and another one is called sexual reproduction. Okay, asexual reproduction will take place by two things, that is by the fragmentation process. Fragmentation means you have the sponges are cut into various pieces and each piece will grow into another. So that is what fragmentation is. Even it is also done by budding. The asexual reproduction is also done by budding. So budding is also called as gemmules formation. So we can even talk about, instead of budding, we can talk about gemmules. So gemulous formation. Gemulous formation means a bud will be formed inside. Actually, so if this is a sponges, it forms a gemulous like structure like this, and so this is having structure. Inside this, it has many kind of buds, inner buds. Okay. Now, when the condition is unfavorable, not fit for survival, the sponge will secrete an outer coat like this. This coat is called encystment. Okay, a covering. So inside this many buds will be there. Now, when the condition becomes favorable, this kind of encystment is broken down and it liberates the buds, it bursts and the buds will come out. So that kind of a formation is called gemulous. When we come to 12th standard, we will study depth about this gemmular formation. Okay. So for the timing, you remember, it is done by fragmentation as well as gemulous formation. Okay. And sexual reproduction is always fusion of the gamete, that we already know. So the reproduction of sponges are, both reproduction takes place, that is the asexual reproduction as well as sexual reproduction. Asexual reproduction is fragmentation as well as gemulous. Okay, right, that is the reproduction part of it. Okay. So now, next one, we will talk about the eye. Eye, here we are going to talk about some 2 to 3 things that we will discuss here. So whenever we talk about eye here, you please remember the digestion part of this sponges. So we will talk about I stands for digestion. Why? Here I am talking about I for digestion is, so you remember whenever you talk about I, one word you put it here, D. So ID. So this word you can remember when you study. It will help you to recall it, that's all. That is the idea of giving this word called Porifera match. Now, what is for D or I? So we will talk about digestion. That is why I told you to remember I. So I is for digestion. Okay. So always the sponge's digestion is intracellular digestion. You study, no? Already we studied conocyte. So the digestion is intracellular. That is why I told you to remember I, D. So digestion is intracellular. One more thing you remember for I, that is fertilization is internal. Internal fertilization. So fertilization will always take place internal, not outside the body, inside the body. Why? Because they are hermaphrodites, then they are asexual, all this. So now this kind of for I, you remember intracellular digestion and fertilization is called internal fertilization. And one more thing you remember, if the fertilization is internal, the development is also indirect. So the development is indirect. indirect development. Indirect development. Okay. So you remember intracellular, internal fertilization, indirect development. So that is the very important thing you remember for I. So I you have, so far we were discussing P for pore bearing animals, O for osteosculum and spongiosum, R for reproduction, I for digestion. So I told you to remember ID so that you can easily remember intracellular digestion, then internal fertilization, then you can also remember indirect development. Okay, right. So this is important point of the digestion is right. So next one we will talk here, that is for fertilization process. Right. So next letter is F. F is for, again we can even talk about fertilization. Already you know the fertilization is internal fertilization. So it is internal fertilization. Fertilization is internal. Even what F is also talks about even the fragmentation process. If you forget here to remember fragmentation, you can even recall there for fragmentation. So F is for internal fertilization and fragmentation. Okay, right. So next letter is E. So E stands for the excretion. E is for excretion. So here the excretion is not given separately. There is no excretory organ. Okay. So excretory system is actually completely absent here when you talk about the excretion. So you do not have any excretory organ at all. So you remember when you talk about excretion, we are talking about ostia. Why ostia? Because through this water enter inside and again it comes out through osculum. Okay, so that is for E. Then R, one more R is there. So any one R you can remember for reproduction, and one R is very important, that is talking about the symmetry. So here the sponges are for radial symmetry. R is for radial symmetry. So radial symmetry means, you know, if you cut, it will give you two halves, but there is only top and bottom, but it do not have the left or right, back or front. So that is what the symmetry called radial symmetry is. Clear? Right, that is for R. Then A. A we will talk here for the habitat. Okay. So A, whenever you talk about habitat, it is aquatic. It is aquatic. Okay. So very important. One more example, one more I'll tell you. When you remember, see, some are aquatic and few are marine and few are freshwater. So there are freshwater sponges also is there. Marine sponges also is there. So they are aquatic. Okay. Now, for A, one more thing you remember, the sponges, some sponges are radial symmetry. Okay. And even some sponges will have asymmetrical. Asymmetry. So that will not give any symmetry at all. Okay, there are sponges. If you can see the diagram part of that in your book, there are different sponges will be. This type of sponges will be there, and one more sponges will be like this. Like this sponges will be there, and this sponges gives you asymmetrical. Okay, so there are, that is the thing that you have to remember. So sponges, they are symmetrical, radial as well as asymmetrical. Is that clear? So we studied all the letters Porifera. So we will complete all the part of it. That is pore bearing animals. They are distinct characteristic we studied. Reproduction, digestion, fertilization, development also we studied. Excretion, then the symmetry, that is either the radial symmetry or bilateral symmetry, all this. Then we have this A for habitat, aquatic or all this. Clear? Right. Then we will talk about MACH. Actually the T I have just removed. So we have for MACH. So that all one we will have it. Okay. So what is C? What is A? M. So we will go with M, letter called M. So M, it is multicellular, primitive, mostly marine. They are multicellular animals. Okay, primitive. What is primitive? First organism. Primitive. Okay, as well as they are mostly marine. Okay, so this part is very important. Few are freshwater and they are mostly marine. Okay, few are freshwater, mostly they are marine. Is that clear? Right, that is one. Then we will talk about A. A is for already I have told you. You can remember even asymmetrical, you can remember. Okay, even you can remember one important that we have missed out here, that is called the role of amoebocyte. So the amoebocyte is where the calcareous shells will be formed. So that point also you have to include. So whenever you write a particular phylum, first you mention the distinct features and then you come to this kind of a method for understanding. Okay, right. So next one is the C. So what is C here? C, you can remember so many words here. C is for canal system. Very important. That is the canal system. Canal system is this one, water canal system. Okay, so that is number one. And you can remember the conocyte, or you can also call as collar cells. So these words and all you can remember easily. You can remember for 1 mark questions. Okay, that is the reason I am just giving. So canal system is there, then collar cells are there, then conocytes are there. Okay, so all these are very important. And one more thing you remember for C, please remember this. Whenever you remember C, this is cellular level of organizations. Cellular level of organization. Very important. Okay. So cellular level of organizations. Whenever you remember C. Right. So what is H? H stands for hermaphrodites. Hermaphrodite. What is hermaphrodite? Both male as well as female sex organs are found on the same individual. Sperm also will be there in that same individual. Earthworm is also an hermaphrodite. Okay, so all these are the distinct, the salient features. So we studied all, that is the subtopics. You remember now, you can just recall it. If you have taken notes, you can just go for the subtopics. We have got, first subtopic is habitat. What is habitat? We are aquatic, mostly marine, and they are freshwater. So this is the first one that we studied, habitat. Second one, level of organization is cellular level of organization. Third one, the symmetry. What kind of a symmetry? Radial symmetry as well as asymmetrical. Okay, then fourth one is coelom. Coelom, it is not there. Okay, so coelom is a acoelomate, so we are not writing. Even if you want, you can write it here. It is acoelomate. Okay, so it is the coelom actually it is absent. That is what it is important. Okay, so whenever you remember symmetry in your NCERT book, it is given as various. Various means it will have radius symmetry as well as it will have asymmetrical. Okay, so various part you remember whenever you remember the symmetry. So it is various. Various means either it is radial symmetry or either it is asymmetrical. Okay, now coelom is completely absent. Then we will go for segment. Segment is not there naturally. So not there means you do not write it. So segment is also absent. Coelom is also absent. Then we will go for the digestion part of it. What is the digestion? Intracellular digestion. Okay, so that is the digestion part of it. Circulatory system, we don't have here particular circulatory system. So circulatory system is absent. Okay, through the water only circulation takes place. There is no organ. Okay, so that is why we have not given open circulatory closed. No, circulatory system is absent. Excretory system is absent. That means they don't have organs to carry it out. So everything is carried out by this throughout this water canal system. So everything, digestion, food gathering, circulation, respiration, excretion, everything is carried out by the water canal system. So all the circulation, excretion, everything is called as water canal system. Water canal system. That is why I just told you here it is the canal system is present. Okay, so we don't have. So excretion part is completely absent. Okay. Then what kind of respiration? Respiration is absent, I told you. Distinct features already we have discussed everything. Right. Sex is hermaphrodite. Then reproduction, when you talk about, we have got the other reproductions like asexual reproductions also will be there, as well as sexual reproduction also will be there. Okay, right. So what is the fertilization? Internal fertilization process. So sexual reproduction either by fragmentation or by gemellus formation. Okay, and what is the development? Fertilization is internal, I told you already. Development is indirect development. Okay, so that's all. So all these salient features we are covered up with a single word called Porifera match. So you remove only the T and you can all remember these words. It will be easy for you to understand and then study. Clear? Right. So now we will do with the example for this Porifera. Example also, I just do it now. Now we will talk about the examples for the Porifera. I will give you a statement for you to remember. Remember the statement called, a very simple statement, Psycho use sponges to do portraits. This is a statement. Portraits, you know, the picture, okay. So, person called Psycho, he uses sponges for what? For doing portraits. So, easily you can remember the examples now. The Psycho is for Psychon. So, Psychon is the first example for the sponges or the Porifera. Psycho for Psychon. Second one, use for Euspongia. Euspongia is the second example. Okay. And sponges for Spongella. Spongella. So these are the examples for Porifera. So easily you can remember. Psycho for Psychon, use for Euspongia, then sponges for Spongella to do portraits. So this is how you can remember the entire examples for the Porifera. Is a simple method for recalling it. Okay. So likewise, if you even study that statement which I have given, Porifera match, you can easily remember all the salient features of this phylum called Porifera. Okay, I hope you understand how can you remember this Porifera now. So next phylum we will concentrate on the Cnidarians. Next phylum that we will discuss is the Cnidaria. Cnidaria is also called as, it is a needle or sting cells. Now, previously this phylum Cnidaria is called as Coelenterata. The old name for this called Coelenterata. Now, before we could study the the distinct features, I will just talk to you regarding this habitat. Separately we will study habitat now. Now the habitats are actually, these Cnidarians are the colonial, in the sense they always remain in the form of colonies. Okay, so they are called colonial. So they remain in the form of a colony, or sometimes they are solitary. Solitary means they are individual. Okay, so they are colony or they are solitary, and they are either sedentary, in the sense they fix to the substrate. They will enter into the aquatic medium and they will fix themselves. So that is called the sedentary. Okay, or they are free living. So either they are free living or they are free swimming. So they are, you can find it, the Cnidarians are either in the form of colony or solitary. Sometimes they are sedentary or they are free swimming. Okay, and they are mostly they are marine, and here also there are some few are fresh waters. Few are fresh water animals. An example for this is called Hydra. So Hydra is an example for, there are so many examples, but Hydra we will discuss here. So Hydra is an example for this kind of a Cnidarians. Clear? Right. So now we will talk about the general characteristics, I mean the distinct features. So the distinct feature is, this type of organism, a Hydra, will have a special type of cells called cnidocyte. Cnidocyte. So cnidocyte is a special type of cell which is present only for these kind of a phylum called Cnidarians. Okay, and this contain a stinging structure. Contain a stinging structure will be present in order to capture the prey. So it is having a structure for stinging. Okay, so the stinging structure is called nematocytes. Nematocytes. So where these nematocytes are present, these nematocytes are present on the tentacles. On the tentacles. Okay, so these are all the distinct features of this particular animal phylum called Cnidaria. Now, the diagram part of this I will explain it so that you can able to understand this stinging cells, nematocytes and all. So here it is having this gastric glands, I mean the mouth part will be there on the top. This is the mouth of this animal. So this is mouth. Okay. And this one, it will be surrounded by a thread-like structures like this. So this is the thread-like structure, and this thread-like structure will have kind of needle-like structures like this for stinging. So this is what the cnidocytes are. Now these cnidocytes will be presented whenever, so this is actually the sting. So this is the structure of the cnidocytes. Okay. So here the cnidocytes will form a sting-like structure, and that has the nematocytes. Okay. Right. So it is touch sensitive. Whenever an organism, something is touching, actually this sting will be present inside in the form of a circular, in the form of a thread. Whenever an organism touches this, the sting will be released. It comes out. Okay. So this is one fundamental features of this phylum called Cnidaria, that is called the nematocyte, number one. Okay. Second one, it is also having a kind of a cardiovascular system, sorry, not cardio, sorry, central vascular system. So if you, this is the structure of, this is the structure here. So here you will have this mouth part. This is the hydra. So this is the mouth part of it. Okay. And this cavity is called central vascular cavity. And this central vascular cavity is for both. Is for digestion. This is for digestion as well as for circulation. Okay. So we have this central vascular cavity for digestion purpose as well as for circulation purposes. Okay. And here this part is called the tentacles. This is the tentacles, and this tentacles only will have this nematocyte, which is the stinging cells made from the cnidocytes. Okay, so that is all very important regarding this kind of a thing. So now what is the role of these tentacles? It will be anchoring, any prey comes, it will be anchorage, anchor. Then even for locomotion purpose also it is being helpful, locomotions. So that is the role of this tentacle. So tentacles helps in the anchorages, then locomotions and all this. Okay. So what is the reason it anchors? It just for capture any prey. So that is the reason it is having this tentacles. Okay. So these are all the particular characteristic for this particular species. Okay. So this is one. And second one, it is having two forms. So this form is called polyp. So this is the first form which is having the polyps, and one more form is the umbrella shape. So this is the other form of it. So this is one form. So we call this form is the medusa. So there are two forms are there in this hydra. One is the polyp and another one is the medusa. Okay, right. So always when you talk about the polyps are sessile, very important, as well as if you see the body, it is cylindrical. Okay, so it is sessile as well as it is cylindrical. And what is sessile? I told you sessile is very much similar to the sedentary. Sedentary in the sense it is fixing. So it is going to attach on the substrate. Okay, so an example here, there are two examples for polyps. One is called hydra and adamisia. Hydra as well as adamisia. So all these are examples for adamisia, and hydra is an example for this polyp type. Okay, right. Just opposite to this is the medusa. What is opposite? Here it is, I told you already, it is umbrella shape, and it is cylindrical, it is umbrella shape. It is sessile, so it is free living. Okay, so these are all the difference between this polyps type as well as the medusa. And here you will have Aurelia or jellyfish. Aurelia is otherwise called as jellyfish. So Aurelia, jellyfish is an example for medusa. So two forms. So first part we studied about the cnidocyte. This is the first one. Second one is regarding this the central vascular cavity, the digestive cavity and circulatory cavity is present here. Okay, and this organism has only mouth. There is no anus. No anus. Okay, so only mouth is present. Only one opening is there. And third one, it is two type, polyp and the medusa part. Okay, so it is having single type of mouth, I told you. So since it is having single type of mouth, no anus, so this part, fourth part we will talk about the mouth. Mouth is single opening will be there. So since single opening, it is for both, one for injection, taking food, and other one is for ejaculation or for the excretion purpose. Okay, single opening for both ejaculation as well as the excretion. Okay, right. So now the this Cnidaria, I told you it is exhibited in two forms. One we have this polyp, other one is called the medusa. So together we call it as this process called metagenesis. Very important word that you have to remember. So here we have this metagenesis. So metagenesis. Metagenesis is nothing but we have polyps and umbrella type. Okay, so that is polyp and medulla. So this is called this metagenesis. Okay, right. So here one more thing that you have to understand. See, one type is called polyp, one type is called medusa. Examples you know, hydra, adamisa, then Aurelia or jellyfish. Okay, so these are the examples that we already studied. Now one more thing you have to remember, I just erase this one and then write, there are one more form in it. Polyps is one form, medusa is one form. One more form that we have it is, that is example is very important, so you have to understand this one, that is called obelia. In this obelia we have this metagenesis process. Okay, so one form is called polyps, second one is called medusa. In obelia we have metagenesis. Okay, individually polyps, medusa, you cannot call it as metagenesis. Together, polyps and medusa together we will call metagenesis. This metagenesis is present only for this obelia. What is this metagenesis? That is, you remember like this. Okay, see, for P M A S. So like that, if you can write it, you can easily remember. So what is P? P is for polyp. In metagenesis means polyp will undergo asexual reproduction. Asexual reproduction to give medusa. M for medusa. And this medusa will undergo sexual reproduction to form polyps. So polyps reproduces asexually by into medusa, and medusa reproduces sexually by into polyps. So this is called metagenesis. It is present only in the oblea. Okay, so these are all the very important distinct features of this particular Cnidarians. Okay, so these are very important, and along with this, two things also I will tell you. It is radial symmetry as well as tissue level of organization. Why tissue level I am talking? Only Porifera will have cellular level. Cnidaria and next phylum Ctenophora will have tissue level of organizations. Understand? Right. So very important. So please remember this one. These things are very important. Okay. So oblea will have metagenesis. Hydra alamisa will have polyps, and Aurelia as a jellyfish for medusa. These are all very important distinct features of this particular animal. Okay. So I hope you understand these distinct features. If you are okay with this, then we will go for another statement which I will give now. Right. So now we will start studying about the salient features. I will give you a statement for remembering. Very simple. You remember a word called merry. Merry, no? You know what is merry? Happy. Huh? Merry Cnidaria. That's all. Simple word. I'll just tell you. Merry Cnidaria. In this, you just leave this word A-R-I-A. We will just take only these letters. Okay. Right. So we will talk about merry. M talks for metagenesis. You know what are metagenesis, no? So metagenesis is for oblea, that is polyp reproduce asexually into medusa, and medusa reproduce sexually by polyps. Okay, you know all this example, all these things. Okay, so this is what important. So it's very important. 1 mark question. Polyp always produce asexual reproduction. Medusa always have sexual reproduction. Okay, individually also examples, you know. Polyps, you will have Hydra and Artemisia, and Medusa, you will have 2 examples. What are the 2 examples? That is, only one important example, that is Aurelia or jellyfish. Okay, so that is called metagenesis. Right. Now E. So the letter E stands for M, we had it for metagenesis, and the letter E stands for extracellular digestion. Extracellular digestion. Why here we have extracellular digestion? Because it is having a digestive tract inside. Only the opening is single opening. Okay, but still it is not there inside the cell. So it is extracellular digestion. And what is R? R I already told you, it is radial symmetry. Okay, then I. I here you will also find a digestion called intracellular digestion. So intracellular digestion. So that means 2 type of digestion takes place in the Cnidaria. One is called intracellular digestion in certain organisms, extracellular digestion in certain organisms. Okay, right. If you can see the book, you will easily understand. Digestion is intracellular as well as extracellular digestion. Right. So now the next one is for C. So C, what are the things that you remember? One is called the cnidoblast. Okay, so cnidoblast, that particular feature that you remember. Second one is central vascular cavity. So this is one. Okay, so cnidoblast, central vascular cavity, and very important in corals. If you see the corals, the outer part is made up of a very hard shell, a skeleton, and that skeleton is made up of calcium carbonate. Calcium carbonate. Okay, that skeletal system is made up of. So it is important point that you have to understand regarding C. Okay, right. Then, so Mary is completed here. Then C I have just taken here, and next one is N. N again it is nematocyte. Nematocyte will be present on the tentacles, and you know the role of the tentacles, everything. Okay, right. The next one is I. Here the I we will talk about fertilization. It is indirect, or it is otherwise called as internal and indirect fertilization. So indirect fertilization. Okay. Then D, development, is also indirect development. Okay, but indirect development, but it is having a larval stage, free swimming larva called planula larva, and this larva is always free swimming. Okay, right. So this is an important 1 mark question in this part of it. Okay, so that's all about your general things that we have discussed so far. So I'll just come back to that 10 salient features for this regarding. So the habitat. So here we already studied the habitat. They may be marine. Already I have discussed previously. They may be marine. Free. If you have taken notes, you can just take it. Marine. Few are fresh waters. Then level of organization we discussed. T for tissue level of organizations. Then symmetry, radial symmetry, that we have it here, radial symmetry. Then coelom, not there, so I have not given, so completely absent. Segment also not there. Digestion, we have intracellular and extracellular, so the digestion here we talk about incomplete. Why incomplete digestion? Because only single opening. You remember? Only single opening, only mouth is present, so incomplete digestion. Intracellular and extracellular. Okay, right. So circulatory system I have not given, so it is absent. Respiration also absent. It is not there. Okay, it will take in the form of through our body surface only. It will take diffusions. And the sex part, reproduction part, and all, it's not discussed in your book, so don't worry. We'll just leave it. And next one, your fertilization. I already told you. It is indirect fertilization and development also indirect. An example, planula larva. Okay, right. So after this, we will talk about the examples. Now I will teach you how to remember the examples for this. See, even if you can study in your book, it is not a problem. But the remembering those examples will get confused, and that is the reason I am just giving a statement for you to remember. Okay. So easily you can remember Porifera, match, all this. Psycho uses sponges to do the portrait. All these are here we will studied about the Mary, Cnidaria and all this. Okay. So here an example that for remember example, first you remember a word called war. Okay, so you remember the word called war. So whenever you remember a word called war, you remember Portuguese man of war. Man of war. So here you have war, so I have just given war here. So we will have Portuguese man of war. So a Portuguese man of war is called Paisalia, is a Portuguese man of war. Okay. So Paishalia is a Portuguese man of war. If remember. Okay. So when you remember war in this. Okay. So you remember war. Example you can easily remember for Cnidaria, Portuguese man of war. Then you remember this statement also, that is Papa, word called Papa, then good morning. So that we use this kind of a statement for remembering when we study, and that is the reason we are giving. Okay. So now in this we have PAPA. So this P stands again for Portuguese man of war. That like that also you can remember. And A for Adamisia, and this A for Adamisia. So what is Adamisia? It's a jellyfish. Sea anemone also comes under this. So here this Adamisia, here we will talk about the sea anemone. Okay, so that is what this Adamisia. Okay, then this P stands for Pennatulae. Important, another one. So pen for sea pen. So Pennatulae is for sea pen. Okay, that is one. Then A, again you remember Aurelia. So Aurelia only I told you it is a jellyfish, and here this is for Medusa. So Medusa example only we have discussed. Okay, Aurelia. Then this part is over. So here I have just taken up to go. Okay, so G-O you remember. You do not remember good. So I made a sentence, that is the reason. Okay. And G-O for Gorgonia. Gorgonia is for G. Okay. And what is Gorgonia? It's a sea fan. Sea fan. Okay. This is one statement. And next one, here morning I have just taken only this M. Okay, M alone you can remember, that is enough. So M for Cnidaria. So this is also another important one. So Cnidaria is otherwise called as brain coral. So these words little tough to be studying, but once if you remember these statements, it will be easy for you to understand. Okay, so these are the phylums. When you remember war, Portuguese man of war called PHYLUM. Then Papa, good morning. So P for Portuguese man of war. A for Adamisia, sea anemone. Then P is for Pennatula, sea pen. A is for Aurelia, Medusa. Go for Gorgonia, sea fan. Then M for Cnidaria, that is brain corals. Okay, so these are all the important thing that you have to remember in the examples. So all these examples are given in your book, so you can also refer this. So this will be helping for you to easily understand. Okay, right.